Hi guys, so in this video we will be discussing glucose metabolism and glycolysis. Now when we talk of the word metabolism, metabolism essentially refers to all those particular processes which are involved in a living organism, in sustaining that living organism and in converting different substances into simpler or more complex forms within the organism. Okay, so metabolism refers to two basic kinds of processes which occur in the living organism's body. Right now, they are generally chemical processes, and these may be either creating processes, that means they create new substances, okay, they make new substances, right? Or rather, to put it another way, they basically make more complex substances. Or the second thing which they can do is by breaking down the existing substances, right? So there are two things that they can do. Either they can create new substances or they can break down older substances into simpler forms, yeah? So when we create new substances in the metabolic pathways, then that creating process is referred to as anabolism, right? That's referred to as anabolism. Whereas the breaking down of complex substances, of these complex substances made in anabolism into simpler forms is referred to as catabolism. So metabolism is made up of two main processes. One of them is anabolism. The second one is catabolism, right? So you can easily remember this because obviously the word kata could refer to something called cut, right? And cutting basically means breaking, right? You're basically breaking it down into simpler forms. So remembering it becomes quite simple because when you cut down substances, they basically break down, and that's what basically makes catabolism. And obviously the opposite of that would be anabolism, that's making new substances. So an example of both of these could be, like, anabolism could refer to photosynthesis in plants where glucose is made, right, which is later converted into starch for storage, or it is taken into, stored as starch. And an example of catabolism could be cellular respiration. That is, you break down your digested food particles to release energy, right? So that's a breakdown process, that's catabolism, right? Although respiration is generally also referred to as an amphibolic pathway because it involves both anabolism and catabolism, or that pathway could be involved in both anabolism and catabolism, but we'll talk about that more when we basically get into cellular respiration. But right now, let's try to understand the basics of metabolism, and that's the overview of the basic concept of metabolism. Now, when we talk of glucose in particular, glucose metabolism is generally a part of the cellular respiratory pathway. And this glucose metabolism, being part of the main cellular respiratory pathway, is of special importance because glucose metabolism, glucose in particular, is one of the main respiratory substrates. And being the main respiratory substrate makes it even more important for us to study its process, its metabolism, because generally when we do take into account the processes occurring in the human body with regards to energy release, glucose is one of those key points which are taken into consideration. We generally measure glucose levels for measuring the sugar levels of the body because glucose is the main respiratory substrate, right? It's the one which is used very often, very commonly. So understanding glucose metabolism is very important at any level, whether it be AP level, whether it be IB level, or your A levels, or it be your medical entrances, your MCAT, or your basic biochemistry, which you study in first year of medicine. So this is going to be something really important for you in biochemistry, in IB, in AP, or in A levels. So it's important that you understand this in a lot of detail, yeah? So let's get on with glucose metabolism. Right now we understand the overview of metabolism. We can now go into glucose metabolism and glycolysis. So let's now talk about glucose metabolism. Let's get back to glucose metabolism. Now, as I told you, we already discussed about metabolism. We already discussed why glucose is important for us to study as a specific example of metabolism. And glucose metabolism is basically a very variable form of metabolism because in different types of cells and different types of organisms, glucose metabolism shows variance. Now, we can talk about two different cases when we talk of glucose metabolism, or we can talk about two different scenarios when we talk of glucose metabolism. Yeah. So one of them is where we have an access to oxygen, right? There is an access to oxygen, right? And the other case is where we have a complete absence of oxygen. There's a complete absence of oxygen, right? So these are two important points for us to note, or two different pathways of glucose metabolism, although they have a common link, but the further progression of the metabolism gets different as we go down from that common link. So that's why it's very important for us to basically categorize them. So we may have an access to oxygen, or we may have a presence of oxygen, because obviously, you know, glucose metabolism is basically a very closely linked concept to cellular respiration. That's why it's important for us to understand why oxygen is involved in this process, because obviously respiration and breathing, they both complement each other, and respiration requires oxygen in certain cases when we need an access to oxygen. That's why we should categorize glucose metabolism into these two kinds. So we may have two pathways which can be followed. Either you have an access to oxygen, or you have a complete absence of oxygen. Please note that I'm using the word complete in particular. So when we have an access to oxygen, then too we have two basic pathways which we may follow. Yeah, either that oxygen presence is quite abundant and you have a complete presence. Yeah, so you have complete presence, or you have a significant excess presence of oxygen, right? So that means you have abundant amount of oxygen. And the second one which you may take up is when you don't have enough oxygen, right? There's a limited supply of oxygen. So that's a limited presence of oxygen. So again, that also kind of breaks down the pathway we're going to follow. That again categorizes slightly the extent to which the pathway we're going to follow. Yeah. So in particular, we don't have really two, rather we have three pathways which we can follow. We either have a complete presence of oxygen, we have an incomplete presence of oxygen, or we have an absence of oxygen. Now, when we talk of a complete presence of oxygen, we refer to this scenario as aerobic condition. This scenario is referred to as an aerobic condition because you have an abundant amount of oxygen, right? And this is generally what the cells of our body as humans carry out. We carry out aerobic conditions and aerobic respiration in general conditions. When we talk of limited presence means you may have oxygen for some time, but then the amount becomes limiting. You don't have enough. So then that's referred to as an anaerobic condition. But this isn't purely anaerobic, it is partially anaerobic. Why? Because you don't have enough oxygen. You do have oxygen, but that's not complete presence. There is a slight limit to the amount of oxygen you have, right? And this is generally present in our muscle cells, because if they do not have enough oxygen during muscular exercise, then they take up anaerobic respiration, and as a result of that, they produce lactic acid. Now, that lactic acid may accumulate in your muscles, and that may cause you pain and cramps. And the third case which we have is absence of oxygen. This is completely anaerobic conditions, right? So this is purely anaerobic conditions. There is absolutely no oxygen involved. So, well, you don't have anything else to do. You just have no oxygen at all. So this is how we basically categorize the different types of glucose metabolism with regards to cellular respiration, which we may take up, right? We do have something called glycogen metabolism, which is closely linked to glucose metabolism, but we're going to discuss it in another video. For now, let's try to understand glucose metabolism and its basic parts, yeah? So we've already looked at the breakdown, the categorization of glucose metabolism, but let's just keep in mind that, as I already mentioned, all these three pathways do have a common link at first, right? Then they go on to branch out as different types, but regardless, they do have a common link. So you could think of it as a tree, and that tree has a common root from which three different branches may arise, yeah? So you can think of it like a tree. I know I have horrible drawing. Please don't comment on it. Thank you. But you have these three beautiful branches. Yeah, I like self-appreciation. So the common root which we have here is what we refer to as your glycolysis. Yeah, and that glycolysis is the common connecting link in all these three major pathways of glucose metabolism. And then we have these three branches which we talk of, which are essentially these three types of pathways. So that may invoke your aerobic pathway, it may invoke partially anaerobic pathway, and it may invoke anaerobic pathway. Yeah. So, or C, completely anaerobic pathway. So the common link between all these three is essentially glycolysis. Yeah. So it is essential for us to understand this common link before we go into the specifics of each type. So we have now an idea of the glucose metabolism process and the types of glucose metabolism which we have at present. But let's first understand the connecting link before we go into the specifics of each type of glucose metabolism pathway.
To glycolysis, right? Let's try to understand glycolysis. So, as we already discussed, glycolysis is essentially the main common link between the three metabolic pathways possible for glucose metabolism. Now, glycolysis is basically made of two words. It refers to glucose, which is essentially the main respiratory substrate of the human body or of many respiratory organisms. And the second one is lysis. So glucose is essentially the main respiratory substrate. Lysis refers to breakdown. So this process is essentially the breakdown of glucose. It's the breakdown of glucose to two molecules of a 3-carbon compound known as pyruvate. So it breaks down into two molecules of a 3-carbon compound called pyruvate. Obviously, why two molecules? Because glycolysis, in glycolysis, glucose is a 6-carbon molecule. So obviously, you cannot destroy atoms, can you? And there's no release of carbon dioxide or any carbon compounds in this process. So both molecules should be 3-carbon, and that's why you need two molecules to balance out the number of carbon atoms. So let's now try to understand the glycolytic pathway. Before we do that, you need to understand that glycolysis occurs in the cytosol or the cytoplasm of living organisms and all living cells. It's a process which is common to all living cells, right? Regardless of whether they are aerobic, anaerobic, or whatever. Glycolysis occurs in all respiratory organisms, respiratory cells, and it's a common connecting link, as I told you. It's the root of that tree with 3 branches. So, cytosol, the cytoplasm, is the site of glycolysis, yeah? And because obviously every cell does have a cytoplasm, it may not have mitochondria, but it does have a cytoplasm. So obviously glycolysis can occur in all living cells which have a cytoplasm, which is obviously every cell. So let's now try to understand the basic concept of glycolysis as well as the main pathway. It has 10 steps. This pathway has 10 steps, and each step has its own significance. So let's try to understand the steps of glycolysis. We will then look into the regulation of glycolysis, and we'll revisit the regulation in a more specific manner in coming videos. So glycolysis essentially starts off with a molecule of glucose. So glucose is your main starting molecule for glycolysis, and you already know that this is a 6-carbon molecule, right? So glycolysis begins with glucose being phosphorylated, yeah? So it becomes phosphorylated, and obviously if you're phosphorylating the process, or if you're phosphorylating a molecule, the source of the phosphate group could be either the cytoplasm, right? But which is very rare. That's only a very few cases would you use the phosphates floating in the cytoplasm. Generally, the main source is obviously the terminal phosphate of the energy currency of the cell, that is the ATP. So ATP would come up and give up a phosphate group to the glucose molecule. So glucose, as a result, gets converted into glucose 6-phosphate. So glucose forms glucose 6-phosphate, and this glucose 6-phosphate has a phosphate group on its 6th carbon. So if you've seen the cyclic structure of glucose, which is essentially this molecule right here, this carbon here is the 6th carbon, and this 6th carbon now has a phosphate group. So this is how this glucose 6-phosphate is formed, right? And there's an enzyme involved in this process which is known as hexokinase. Hexokinase enzyme is what is involved in converting glucose to glucose 6-phosphate. It's a type of phosphorylase which phosphorylates the molecule. We then have glucose 6-phosphate, which then gets converted into a fructose 6-phosphate by an enzyme known as isomerase. So this is a type of enzyme which carries out the isomerization of certain molecules. And you already know that fructose is the isomer of glucose. So that being said, it's quite obvious that obviously isomerase would be the enzyme because you're isomerizing glucose 6-phosphate. But what's the purpose? Why do you isomerize it? Why don't you just carry out the metabolism through glucose 6-phosphate? Well, the answer for that lies in that fructose 6-phosphate is relatively more reactive towards certain biochemical processes. And that's why it's important for us to convert this glucose 6-phosphate to fructose 6-phosphate so that it becomes easier for us to carry out the downstream reactions of glycolysis. We then get started with fructose 6-phosphate being converted into fructose 1,6-bisphosphate. So fructose 1,6-bisphosphate. Bisphosphate means the phosphate is not on the same carbon. They are on two different carbons which are not adjacent to each other. So 1,6-bisphosphate means there's a phosphate on the first and the sixth carbon of the fructose ring. And the enzyme involved in this process is it's a type of phosphorylase, and that's called phosphofructokinase 1, PFK1. Phosphofructokinase 1 is the enzyme involved in phosphorylizing fructose 6-phosphate to fructose 1,6-bisphosphate. Yeah? And again, since you are phosphorylating the molecule, there needs to be a source of phosphate, and that source is yet again ATP. So ATP gets converted into ADP, and that phosphate group is given to this fructose 6-phosphate to convert to fructose 1,6-bisphosphate.
Fructose 6-phosphate will then get converted into obviously, it does get very unstable, has a lot of strain, two phosphate groups. So fructose 1,6-bisphosphate then breaks down into 3 carbon molecules. Now remember, till here you have 6 carbons. All of them have 6 carbons. But here, it's quite unstable. And this fructose 1,6-bisphosphate then breaks down to give rise to one molecule of glyceraldehyde 3-phosphate, GAP. Glyceraldehyde 3-phosphate, and another molecule of dihydroxy acetone phosphate. And remember that these two molecules, since there's a cleavage of the 6 carbon molecule and atoms cannot be destroyed, so each of them has a 3 carbon content. So each of them has 3 carbon atoms. So they're all referred to as, they both are referred to as triose phosphates. They both are called triose phosphates because they both have 3 carbons and a phosphate group. So one phosphate group here, one phosphate group here, and 3 carbon atoms in each molecule. So they're called triose phosphate, right? Now, this cleavage now brings us to the next phase of this reaction. Now, why do I use the word phase? Because till now, whatever you've seen in this particular part of glycolysis, you may notice that this particular part of glycolysis involves a lot of energy investment. As you can see, there's a lot of use of ATP. There's ATP being used at every step, a lot of steps here. There are 2 ATPs already used up. So this phase involved the use of a lot of energy, and that's why it's called the energy investment phase. So once we're done with the energy investment phase, we then move on to the energy recovery phase. And that's what we talk about after GAP and TAP. So obviously, there's one important point here, that obviously you have 2 molecules of each thing being produced. So that means you need 2 molecules of GAP to proceed so as to make sure that you have 2 molecules of pyruvate at the end. So that's ensured by the isomerization of GAP and DAP. So DAP gets converted into GAP, right, through isomerization because they are essentially isomers of each other. So DAP, that is dihydroxyacetone phosphate, gets converted into GAP, that is glyceraldehyde 3-phosphate. And that's basically one of the reactions of glycolysis. It's counted as one of the reactions of glycolysis, right? So till now, how many reactions have we completed? That's 1, 2, 3, 4, 5 reactions. So we have 5 more to go because there are 10 steps. So let's now talk about what happens after we have 2 molecules of GAP, one coming directly from fructose 1,6-BP and the other coming from DHAP. Let's now go on to see what happens after we have 2 molecules of GAP. So when we have 2 molecules of GAP, as glyceraldehyde 3-phosphate, right? What happens after we have these 2 molecules of GAP? So after we have these 2 molecules of GAP, that glyceraldehyde 3-phosphate gets converted into 1,3-bisphospho glycerate, right? So as you can see that there is an addition of another phosphate at the first carbon because this one already had a phosphate at the third carbon. So what's the source of that phosphate? Again, you would think that it's ATP, but we just finished off the energy investment phase. So there can be no more use of phosphate groups anymore. So now we need to, no more use of ATP anymore. So what's the source of phosphate? So if you remember, I already mentioned once that the source of phosphate could be the cytoplasm. And that cytoplasm is the source of the phosphate for this reaction. So simultaneously, it picks up a phosphate from the cytoplasm. And simultaneously with that, at the same time, there is an NAD plus molecule which comes up. Now, NAD plus nicotinamide adenine dinucleotide is a cofactor which can accept electrons. So it accepts electrons and gets reduced to NADH, right? So obviously we are using two molecules of GAP, so there would be two such molecules being reduced. So NAD plus gets converted into NADH and a phosphate is picked up from the cytoplasm to form 1,3-bisphosphoglyceric acid or 1,3-bisphosphoglycerate. Then on, we start making energy. So we already used two energy molecules, ADP. Now we start making energy. Again, we have two molecules of 1,3-bisphosphoglycerate. Now, an ADP, that is adenosine diphosphate, picks up a phosphate from this 1,3-bisphosphoglycerate and forms an ATP molecule. So now energy is going to be made. Now we're making energy. Like now we are back at the energy recovery phase. So it picks up a phosphate and it forms a molecule of 3-phosphoglycerate. And that to two molecules, because obviously you're making two molecules of everything. So two molecules of 3-phosphoglycerate. This two molecules of 3-phosphoglycerate then undergoes an isomerization reaction where a phosphate group moves from the third carbon to the second carbon, and this forms two molecules of 2-phosphoglycerate. Right? So that's making two molecules of 2-phosphoglycerate. It basically changed its position from the third carbon to the second carbon of the glycerate. So then on, again, there is the addition of water. Now, this is something new. There's a hydration reaction occurring. There's a water molecule which is going to enter this process, and it's going to form something called an enol. Now, you know what enols are. Enols are those those organic molecules which have an OH group on a double bonded carbon. So this is what we call an enol. Now, this is how we're going to form this enol. We're going to add water molecule so that it forms two molecules of 2-phosphoenol pyruvate. Right? So that's going to form two molecules of 2-phosphoenol pyruvate. So we're already at pyruvate. The only thing which is stopping us is this water and the phosphate group. So at the last step of this reaction of glycolysis, an ADP is going to come by. And that ADP is going to accept a phosphate from this phosphoenol pyruvate and form ATP. And that's going to give rise to your phenol pyruvate. But again, water will leave simultaneously as this phosphate is picked up, and that will give rise to two molecules of pyruvate. And that's what we needed in our final product. So just see here, we started off with two molecules of GAP in this phase, reached two molecules of pyruvate. We have 5 reactions occurring, 1, 2, 3, 4, and 5. And we needed 5 reactions because the total was 10, and we have 5 reactions. Now, can you make the balance sheet? Let's have a look at the balance occurring in this. So in the balance sheet, you can see there is 2 molecules of NADH being formed. Why are we multiplying with 2? It's because there are 2 molecules of GAP. Similarly, we have 2 ATP forming here and 2 ATP forming here. So we have 4 ATP being formed in the energy recovery phase. So that's the 2 main things to note. But you also need to keep in mind that in the energy investment phase, we used 2 ATPs as well, because we used 2 ATPs to give phosphates, right? So what's your net gain? The net gain is 2 ATP. So this is your net gain. So the net gain of glycolysis is you have two molecules of ATP being formed together with two molecules of NADH. Now, why are we giving importance to this NADH? Because it is also an energy reservoir. It's going to store energy and going to release free energy in the last step of aerobic cellular respiration in the electron transport chain, which we will study later. But right now, try to understand that there are two net ATPs being formed together with two NADH molecules being formed. So that's the net gain in glycolysis, a 10-step beautiful process. It may look complicated. You can see there is a lot of stuff going on, right? There are 10 steps. But regardless, you don't need to worry about that. You just need to make sure that you understand the basic concept. That is, at different points, what is the exchange? What molecule is being exchanged? What molecule is changing, right? So you need to understand glycolysis in a more broader sense rather than just looking into specific steps and memorizing or rote learning. And anyways, if you write it a few times, it just sticks to your mind, so don't worry, all right? So that's about your glycolytic pathway. That's the basic glycolytic pathway. In the next video, we will be discussing the regulation of glycolysis. That is, at what steps is the most important change in energy, which leads to a lot of changes in the glycolytic pathway or in the direction of the movement of glycolysis. And which steps are those steps which can make that happen? So stay tuned and do like, share, and subscribe to my channel. Goodbye.